I am David Miller. I'm the co-director of the NF Clinic at Boston Children's, and I'm here with Dr. Ulrich, who's the other co-director. I think it's on, but try, or we'll share. I just want to say first off, well, thank you so much to Dr. Plotkin and the MGH team for doing such a great job again with putting on this wonderful event. I always enjoy coming here because they do such a great job. We have the distinct pleasure of introducing Dr. Bruce Korf, who is one of the leaders, the international renowned leaders of NF and schwannomatosis related research and clinical care. He pretty much has done everything for the community of NF, his tremendous dedication to patients and research and clinical care. So there's not, I could go on forever. We're talking about his accomplishments, but we're just so fortunate to have him here to give the other keynote speech.
It's great to see so many familiar faces. In 1983, Dr. Korf started the NF program at Boston Children's, and at a previous occasion, I shared the announcement of that clinic. He was only a fellow, not only, but a fellow in neurology and genetics. So that was kind of a bold move, and there was a typewritten newsletter that came out at Boston Children's. And both David and I had the opportunity to his footsteps are large, so we maybe took a little piece of that, but to follow him in leading the program at Boston Children's and have also benefited greatly from his mentorship along the way. So we hope to carry on the torch and the legacy that he's left us and share that with you. So without further ado, Dr. Korf.
Well, Nikki and David, thank you for that introduction. Scott, thanks very much for inviting me. Brad, we have indeed spent many an hour around conference tables over the years. It is really wonderful to be back here after 21 years in Birmingham, which was a great experience, but it's really wonderful to be back. So Scott said I could speak about anything I wanted, as long as it was about NF. And so I decided with 42 years, as you've just heard, to use the time to reflect a little bit. And so what I will do, let's see which button you press here. Is it this one?
We had a little pointer.
It doesn't matter which.
Yeah, it's down this guy right. You want the pointers right here.
Okay, but how do you make this go to the next? Oh, I see.
Yeah, there you go.
Perfect. Okay, thank you. All right, so I thought I would do these four things. First is say a few words. Nikki's already alluded to it. I'll elaborate a little bit on how I became involved. Second, what's changed in the 42 years? A lot. I'll just pick out a few things. Third, just comment on some things I think I've learned along the way. And finally, a look into the future. So let's see. Yeah, so you just heard about the clinic having been established at Children's in 1983. These are the two people who made that happen. Park Gerald is the person on the right. He was the division director in the division of genetics at the time. I was a fellow, as you just heard, training in genetics and neurology. So he called me into his office one day and said, look, you're training in genetics and neurology. We have this condition that affects the nervous system and is inherited. And Joseph Murray, that's the person you see to the left. He was the head of plastic surgery at Children's at the time. And his team was doing surgery on people with NF, but nobody was really looking after the kind of big picture care of people with NF, and that the surgeons weren't really equipped to do that. So he asked Park Gerald, is there anybody in your group who might be willing to take this on? And at the time, maybe I had seen a handful of people with NF in my training. And I actually had attended a lecture, probably within a few months of that time. It was given by somebody whose name may not be known to most of you, Judy Bader. She was one of the co-founders of the National Neurofibromatosis Foundation, as it was known at the time. I don't think she stayed in NF over the long haul, but it was a really interesting talk. And note to colleagues, you just never know who you're going to influence when you give a talk. And so I said, sure, I'll do that. And this is the announcement that was put out, and it was actually sent around, and Dr. Murray then sent it back to me saying how glad he was that we were doing this. I actually met him in his office, and he had the somewhat intimidating sort of habit of dictating a note about the meeting while you were still there. Small parenthetical. It turns out, I didn't know this at the time because I knew him as a plastic surgeon. He did the first renal transplant and got the Nobel Prize for that several years later. I had no idea when I met him. So there was one more piece of the puzzle that will not make sense to most people probably. Why show a corn cob here? The person pictured is Barbara McClintock, who was a geneticist who worked in the 1940s on a phenomenon in corn where you can see the sort of spottedness of the kernels. And when I was a graduate student, so that's now into the 1970s, really just a few years before the clinic was started, it was discovered that the reason this happened, something she wouldn't have known because actually DNA structure wasn't even known at the time when she did this work, but it was unbelievably meticulous and really beautiful piece of work. It turned out the reason this happens is there are what some people call jumping genes, little chunks of DNA that can move around from place to place in the genome. And if they land in a region of the genome that encodes pigment, it can disrupt the pigment. I was really fascinated by that as a graduate student. So here I was now seeing people who had skin spots, and I thought, well, maybe it will turn out to be that. It turned out it wasn't. That's not why cafe au lait spots happen. There are some NF1 mutations that are the result of these jumping genes, but it's a fairly small minority. I'll say more about that in a few minutes. All right, so what's changed in the time since that clinic was begun? And a lot of things, of course, have changed. I'll just pick a few. One of them is imaging. So when we started, there was no such thing as MRI. We used CT scans. I didn't actually have one of my own to show. That's why I pulled this off the internet. You can see the little credit, or maybe just barely with the captioning. But in any case, those were the kind of fuzzy pictures we had to work with to diagnose things like optic pathway tumors back at the time. I actually remember the first time I saw a brain MRI. It was done here at MGH. There was an experimental unit at the time, and I was just amazed at the anatomical detail that you could see. This is a more modern day picture showing bilateral optic pathway tumors in a person with NF1. So imaging improved dramatically, and also then the ability to do volumetric MRI. This is a plexiform neurofibroma shown at the right, and the green outlines the boundaries of that tumor, and that permits the volume to be measured. So it provides a really accurate way of assessing the rate of growth of the tumor. So actually that work was done starting in the 90s, spilling over into the early 2000s. It resulted in the ability to put together growth curves. And what you're seeing at the left shows that most of the growth of plexiform neurofibromas happens at a relatively young age. And not that it can't grow later on, but the growth is a lot slower than it is in children. Well, what about diagnosis? So this is a paper that was published in 1992. It was the first paper I ever wrote on NF, and it's the last paper I ever wrote where I was the only author, I should say. Yeah, it was a simpler time. And so we did a study just looking at if a child showed up with cafe au lait spots, what was the chance they would actually eventually develop NF1? And the answer was high, not 100%. We didn't know about Legia syndrome, this other condition that causes cafe au lait spots, and there are many others too. The diagnostic criteria that we use today were put together in 1987 at an NIH consensus conference. A very kind of, I don't think they have them anymore, but it was a kind of interesting experience. You would go, and I was there, you would give a talk about whatever it is you were working on, and there would be this jury, literally, there were, I don't know if it was 12 people, but there was a group of people at the front of the room who would listen, generally they were not NF experts. They were experts in neurology and genetics and the like. And their job was to take all the information that they heard and distill it into something that would provide sort of guidance for future care. And among the things that came of that were the diagnostic criteria, which really did stand the test of time. And so, in fact, they were revised, and Dr. Plotkin was a big leader in that effort just a few years ago, in the same sort of breath as where the nomenclature that he alluded to earlier was modified. And the diagnostic criteria were tweaked, I think it's fair to say. They're still not too different from what they were. The ability to do genetic testing, about which I'll say more in a second, was added to the list that reflects the fact that there was no genetic testing for NF1 back in 1987. And so what about genetics? So in the upper left is another paper that I was able to participate in. It was kind of a remarkable experience in that this would have been around 1987, as I recall. The gene for NF1 had not been located yet. And a group of scientists, not including myself, got together and compared notes as to where they had looked in the genome and not found the gene. And when they did that, they discovered there were just two gaps that hadn't been looked at, and one of them was on chromosome 17. So a group from MGH, Jim Gazella and Bert Seisinger, came back and said, it's got to be in one of these two places, and we need more families in order to be able to do the genetic linkage studies. So we had a family that we were planning to work with to ultimately enroll them in a study. And he said, well, how quickly can you do it? We went out on a Saturday. They made DNA that Sunday. They did the genetic linkage studies by Tuesday, and the paper was written by Thursday. It was unbelievable just how quickly that happened. And sure enough, the gene turned out to be on chromosome 17. Turned out, though, that it wasn't so easy to do genetic testing. The gene itself was identified by groups both in Utah and back then in Michigan in 1990. But it turns out to be a pretty big gene, not the biggest known, but a big gene, and a difficult one to do mutation analysis in. I moved to UAB in 2003 and was fortunate to be able to recruit a colleague, Dr. Ludwig Messiaen, who unfortunately passed away about a year and a half ago now, and really was a pioneer in developing a practical genetic test for NF1 and actually also for NF2 and the other forms of schwannomatosis. In fact, she was the discoverer of one of the LZTR1 related schwannomatosis. So what you're looking at on the right is a map of the gene. It's a little small, but you get the idea that there are lots of different mutations. Every single mutation is indicated by a star below the line, or those purple horizontal lines are deletions, and the vertical lettering are mutations that affect the processing of the gene. Last count, something like 3,000 different pathogenic variants have been found. And there isn't a lot of correlation between where these variants are or what they are and how severe the NF is. There are a few exceptions to that. 3 to 5% of people have a deletion of the whole gene and usually get a more severe phenotype as a consequence. The pie chart shows the different kinds of genetic variants, and they have in common, through different mechanisms, that they usually turn off the gene. They cause it not to function at all. And I'll come back to that point because it's important in terms of pathogenesis. And you'll see this pie chart again in a few minutes when we talk about some of the work that we're doing, trying to look at different ways of treating NF1. But Ludwig did develop a lab that I think to this day remains the kind of gold standard lab internationally in NF diagnosis or genetic diagnosis. Though I will say these days, technology has changed and it's a lot easier to do that testing than it was back in 2003. And then finally, a change that I think is probably the most important of all is the ability to do treatment. It turns out what's going on in NF1 is that a cellular signaling pathway goes awry. It's called the RAS pathway. RAS lives at the inner cell membrane. when a signal comes to the cell, basically telling it to grow, RAS is turned on. It sends signals down many different pathways, only one of which is shown here. And NF1, in a nutshell, sort of is the off switch, tells the cell enough, you don't have to grow anymore, and turns things off. Well, that works fine as long as NF1 is there. When it's not, which is what happens in tumors, about which more in a minute, then the cell doesn't get the signal to stop growing, and hence you wind up with the tumor. And lots of work over the years looking at ways of blocking that pathway resulted in a family of drugs called MEK inhibitors that in mouse models were shown to reduce the growth of neurofibromas that could be made to occur when the NF1 gene in the mouse was knocked out. And this has resulted, as I'm sure most of you in the NF1 world at least are aware, of now 2 FDA-approved drugs, mirdametinib and selumetinib, that are available for the treatment of plexiform neurofibromas. The picture at the bottom is a patient of mine. I guess I could say at this point a former patient of mine since retiring. But when I met him, he was a young child, and you can probably see, it's a small picture, I know, this really large tumor in his neck. And he was headed towards a tracheostomy because of compression of the airway. And the growth curve at the right shows you the kind of steady growth of the tumor. We were able to get him enrolled at an NIH study, the kind of original study with selumetinib. And you can see in red to the right the shrinkage of the tumor. That was actually just after a year or so on treatment. And he's now been on treatment for 10 years or so. And the tumor reduced in size by about 50%. It's not gone, and you can still see it on imaging, but you actually have to know where to look to see it clinically. And actually, he should be finishing his first year of college at this point. So huge advance in the ability to treat these plexiform neurofibromas medically, because surgery was never a very good option for them. And the fact that there were 2 drugs is a huge step in the right direction. Not the end of the road, however, because first of all, when the drug is stopped for whatever reason, and it does have side effects, either of them do, the tumors tend to grow back. So they're not totally gone. They're suppressed, which is great, but it does mean you have to face what may turn out to be a lifetime. We don't really know at this point, certainly a long period of time on treatment. And the tumors don't go away completely, and not everybody responds equally. So there's plenty of room for further development. On the left side, you see a chart outlining the clinical trials that have been done through the NF Clinical Trials Consortium. So I was the principal investigator of that for about 17 years until last year, when kind of on a trajectory towards retirement, was able to turn it over to a colleague at UAB to run the operations center. But you can see lots of different clinical trials for NF1 and NF2 related schwannomatosis as well. Those of you in the NF advocacy world are aware that in this current kind of financial climate, or governmental climate, really, the DoD CDMRP program in NF has been curtailed, endangering the NF Clinical Trials Consortium. Hopefully advocacy will lead to some change in that outcome. But I think for the moment, the consortium is able to keep going because there were funds already allocated. It remains to be seen, I guess, where this will take us in the course of the next year or so. All right, so a few reflections on things I think I've learned over the course of this 40-odd years. First is resilience. One thing that you just have to be impressed by as a clinician is working with people who are dealing with things that could be at various points in time relatively mild, or could be at other points in times very severe, even sometimes life-threatening or disfiguring. And the ability of people to go about their lives in spite of the many things, that challenges that they're living with, just can't help but to leave you in awe, really, as a clinician, and to realize the ability of people to adjust not to say that they like the situation that they're facing, but that they can still manage to function has always been, to me, a source of inspiration. Second thing is hope. And, so the gene for NF1 was discovered in 1990, and the first FDA-approved treatments were in, I guess, 2020, I think it was. So 30 years passed from the time the gene was found to the point where a treatment evolved. And actually, it's interesting to note that the time from when the gene was found to when there was a pretty fair inkling of function was actually measured in days to weeks, because it became obvious immediately once the gene was known and the sequence of the protein that the gene encodes was known, what it basically did. And although there's been plenty more learned in the decades since, it wasn't long at all to figure out that. What was hard was figuring out any way to deal with it. And that took a very long time. I would hope now, as technologies have improved in terms of drug discovery, that that trajectory will accelerate, not just for NF, but for many other kinds of things. But it is a bit sobering to realize, but it was research that was the source of hope, and it was advocacy that made a big difference to keeping that research flame alive. And then the third thing is what I would call urgency. And this is something I try to explain to trainees, and that is, so you're in clinic for a day and it's a busy day and you see people with, well, whatever it is you're working on, NF in this case, and then you go home and hopefully you're not just doing clinic notes while you're home, but you may do other things and you can kind of leave that behind you for a little bit. Your patients take NF home with them, and it's never not with them. And in my mind, that creates a sense of urgency, that it can never be soon enough to be doing things that will make a difference in terms of the lives of people with this disorder. Now, I will admit that, life gets in the way of urgency sometimes. So it's not like I've always lived up to that myself, but I think that is really the ideal. I think that, a sort of physician scientist working in a field like this really needs to be cognizant of. So finally, just a few words looking towards the future. And this is a kind of a flow chart of what we think is going on when tumors form. So person with NF1 is born with one copy of the NF1 gene. Remember, we have two copies, one inherited from each parent. One copy is damaged by a genetic change. The other should still be functioning. The damaged one is shown by the minus sign and the functioning one by the plus sign. Now, maybe that itself is enough to upset some aspects of brain function, which may be why the learning disabilities occur. I don't think that's been proved, but I think it is a plausible sort of way to look at it. And there is work going on trying to rev up the function of the remaining non-mutated copy of the gene to see if it can be made to compensate. So that's one avenue of at least possible treatment. To get a tumor, the other copy of the gene needs to also be mutated. Now, you may say, well, that would be a kind of an unlikely thing, but in fact, genetic changes are happening all the time in all of us and in cells all the time as they divide. And having one copy of the NF1 gene already damaged puts you at risk of tumors, because if by chance that other copy is lost just by a kind of random accident of cell division, there's no backup copy, so to speak, and that seems to be what triggers a tumor to form. It's not clear exactly how you could prevent that from happening. There may actually be ways of looking at that I won't go into detail about for the moment, but that seems to be right now the rate limiting step. And then once that happens, as I mentioned earlier, the RAS pathway is revved up more than it should be. And the MEK inhibitors I mentioned, those are really aimed at trying to block the RAS pathway. And that's where the success has been so far. There seems to be crosstalk between the cells, Schwann cells in the case of neurofibromas, and other cells that contribute to the tumor. So there may be opportunities to block that signaling, all towards blocking the formation of a tumor. But there's one other potential form of treatment, and that is either restoring function to the mutated gene or gene product or replacing it entirely. And that's the tack that our group at UAB has been taking. So you saw this pie chart earlier, and it basically depicts the different kinds of mutations. And we have what are called missense, which means that the wrong protein building block, amino acid, is inserted into the protein because of an error in the genetic code. And there are examples, not in NF right now. Cystic fibrosis, another genetic disorder you may have heard of, can be treated using drugs that overcome the kind of dysfunction that a missense variant causes. And so one of the things the group is doing is looking for what are called corrector or potentiator drugs. About 21% are what we call nonsense. That's like putting a stop sign in the middle of a street instead of at the end of the street, and traffic backs up behind it and doesn't know where to go. There are drugs that will cause the cell to read through the stop sign, and those are now being tested. The NF gene is complicated, and there are multiple segments, and we're interested in whether you can skip over a segment that has a mutation and still get a reasonable functioning gene, and so that's another possibility. There are mutations that affect splicing, which is part of gene processing, and we actually have had some success using an approach to essentially correct a splicing defect, at least in one instance. And finally, the ultimate might be something that Brad referred to in NF2, and certainly is true in NF1 as well, which is either doing DNA or RNA editing. You probably have heard of the CRISPR-Cas9 system. It's used all the time to create NF in like animal models. Can it be used to correct NF? It's a lot of challenge to do that. There have been gene therapy approaches like in sickle cell anemia, where you can change the cells and put them back in the person with sickle cell and correct the ultimate genetic defect. Problem is, sickle cell is a blood disease, and it's fairly accessible to replace cells in the bone marrow. NF1 affects Schwann cells and many other cell types all over the body, and targeting them is much more challenging. But that is an area that our group and others as well are working toward, or just simply replacing not maybe the whole gene, but at least a part of it that seems to be most important. And again, that's an area of active research. So where are things headed over the course of time? I would mention three things I think that may be the future of NF care. One I call smart surveillance. Does everybody need to be monitored for all the same complications? Can we tailor it to the individual? And although I mentioned earlier, there aren't a lot of genotype-phenotype correlations. In other words, it doesn't matter what your mutation is. Mostly, you sort of get the same outcomes. But what we don't know is, are there other genes that modify the way NF behaves and explain why one person with a particular mutation experiences a lot of tumors and someone else doesn't? So I think there's plenty of room now for trying to refine our approach to surveillance to be more sort of personalized, I guess you could say. The second is targeted treatments, the right treatment at the right time to the right individual. And as we learn more and more about how to develop targeted therapeutics, I think NF will benefit. And then finally, would emphasize the importance of community building, support among members of the community, having people be empowered to participate in data collection. And I think one of the biggest challenges nationally, not to say internationally, is access to care. There are 2 NF clinics here in Boston and none in most parts of the country, never mind the rest of the world. And how do you kind of bridge that gap? And I think they're, short of expecting NF experts to be populating everywhere, there may be AI-based approaches that can really help in disseminating the knowledge so that it's available much more widely. So I'll end with just a few words of acknowledgement, not naming individuals because there are frankly so many I was sure I'd forget some. The ones that started me on the journey, whose names I did mention earlier on, many scientific collaborators over the years, internationally here in Boston as well as at UAB. I've been really fortunate to be associated with colleagues, physicians, genetic counselors, and nurses, initially here in Boston at Children's, as you heard, then later on at UAB in Birmingham. And they've been instrumental in helping things to keep moving. Funding, especially from the Children's Tumor Foundation, NIH, and hopefully continually, but certainly it has been the case from the Department of Defense. And then lastly, of course, the many patients and families who are living with NF and are the source of inspiration for all that we do. And with that, thanks very much.